your childhood just got rewritten. Scientists discovered how to flip genes on like a light switch without cutting DNA. And this changes everything. CRISPR gene editing has been making headlines for years. It's the tool that lets scientists edit our genetic code to fix diseases. But here's the problem. Traditional CRISPR works like molecular scissors, cutting your DNA to make changes. And every time you cut DNA, there's a risk of cancer, a risk of unintended mutations, a risk that could follow you for life. That's why what just happened at the University of New South Wales is a game changer. Researchers created a third generation of CRISPR called epigenetic editing. Instead of cutting DNA, it targets tiny chemical tags called methyl groups that sit on top of your genes. Think of these methyl groups like dimmer switches. They control whether genes are on or off. For decades, scientists argued about whether these tags actually controlled genes or were just leftovers. This breakthrough settles the debate. They are the control mechanism. Here's how it works. The team uses a modified CRISPR system to deliver special enzymes that remove these methyl tags from silenced genes. It's like lifting the brakes off a parked car. Suddenly the gene can activate again, but nothing gets cut, nothing gets damaged. No scissors, no risk. The most exciting part? They can use this to treat sickle cell disease. When you're born, you have a fetal blood gene that works perfectly, delivering oxygen throughout your body. But as you grow up, that gene gets switched off and replaced by an adult version. For people with sickle cell, that adult gene is faulty. It creates misshapen blood cells that cause chronic pain, organ damage, and a shortened lifespan. With this new technique, scientists can simply turn the fetal gene back on. No cutting, no replacing, just flipping the switch. And because there's no DNA cutting involved, the cancer risk drops to nearly zero. This isn't just theory. The research was published in Nature Communications in July 2025, and it's already being tested in labs. The technique could be ready for human trials within a few years. But it gets even better. This same approach could work for hundreds of other genetic diseases. Any condition where you need to wake up a sleeping gene instead of cutting out a bad one. We're talking about muscular dystrophy, certain cancers, metabolic disorders. The list goes on. The researchers say this is just the beginning of a new age. They can remove methyl groups, add them, or target other chemical markers. It's like we just discovered a whole new control panel for the human genome. Now let's talk about what's happening right now in CRISPR medicine. Just 2 days ago, MIT announced they made prime editing 60 times more accurate. Prime editing is another cutting-edge technique, and they've reduced error rates from 1 mistake in every 7 edits down to 1 in every 101. That's a massive leap in safety. And here's something incredible that just happened in 2025. A baby named KJ became the first person ever to receive a fully personalized CRISPR therapy. He was born with a rare metabolic disease called CPS1 deficiency that would have killed him without treatment. Within 6 months, doctors at Children's Hospital of Philadelphia designed a custom gene therapy just for him. They delivered it in February 2025, and today, he's thriving. This is what the future looks like. Gene therapies designed in months, not years. Treatments customized to your exact genetic code. And now, with this new epigenetic editing, we can do it all without cutting DNA. The CRISPR revolution everyone predicted? It's finally here. We're not just editing genes anymore. We're learning to conduct the symphony of life itself, turning genetic diseases from death sentences into fixable problems. And the best part? This technology keeps getting safer, more precise, and more accessible every single month. If you found this mind-blowing, hit that subscribe button. We cover the biggest breakthroughs in science and technology, and trust me, CRISPR is just getting started. Drop a comment. What disease would you want cured with gene editing? See you in the next one.